I certainly hope so. So I would obviously like to start, first of all, before starting with the inflammatory section of my talk, with just a little bit the cornerstones of the company that I'm working for. I belong to the company in.vent Diagnostica from Berlin, Germany, or nearby Berlin. We are ISO certified according to the ISO 9001 and 13485, so according to two different standards of quality management. So we are really taking this seriously. This is because quality and everything related to legal and ethical correctness are like the bread and butter of IVD work in the EU, and this is like the foundation, the groundwork of our services. There, two. Okay. So a couple of surrounding facts about our company is that we are, depending on how many affiliative companies you count in, we are at the basis of around 80 employees with a very high percentage of academia background, which is necessary for our work. We only, I want to stress, so far have about 2,000 square meters of office and 1,000 square meters of lab space, of storage space. But you can basically double these numbers already, because we are expanding right now into a lot of new office space, laboratory space, and we are opening up a complete cold storage building for our samples and for our products. So we are really on the in a growth phase currently. And actually, we have been basically for the last 23 years, and these 23 years are also it's a considerable number in the field. And during all this time and this period, we have set up strong collaborations and very good trust in stakeholders from all parts of the industry, from all parts of biotechnology, medical science, and IVD. And it gave us an excellent reputation, both nationally and internationally. Let's say the majority of our income as a company is coming from procuring and selling human biomaterials of all kinds and ranges. This is like small scale and highly with samples with a high clinical definition, up to just normal human plasma of tens of thousands of liters of range. So everything between this range, everything of this scale can be procured and sold by us. And this includes all kinds of sample types, all kinds of clinical fields, and pretty much for all applications as well. If you need it processed in any way, if you need it aliquoted in any way, this can all be done. So to put it in a nutshell, kind of like tailor-made human biomaterial procurement for the IVD industry. Something that I would like to stress is, since there are also a lot of other biobanks out there, and I would say that we are a kind of biobank after all, a major difference is that usually there is no middleman included in our sample procurement efforts. We ourselves are the ones that place the needle and draw the samples. And we can do this since we have our own collection site, our own study site, a donation center in which our patients that we know, patients with defined clinical conditions, come and donate just for our purposes. And if these sources do not suffice, we have very dense network of very well-established collaborations with clinics and hospitals and laboratories, so we are fit for all purposes. And now, since the goal of my talk here is to kind of shed some light on thoughts about clinical study, design, and especially the sample procurement issues behind this, let me introduce to you also another part of my company, like in.vent Diagnostica, so to say, the mother company. And here we now have in.vent Clinical Services, of which also I am a part. And the function here is that in.vent Clinical Services, or ICS, is kind of a CRO department within in.vent. And the thought behind this is that in.vent procures the samples and ICS puts them to use in clinical performance studies usually. And the logic behind this is obviously that due to the transition to the IVDR, which you all know about, there's a much higher requirements for technical documentation. Clinical evidence is required for basically anything that you do, and therefore there is a high need for clinical performance studies and also consulting and services regarding this. And this is also the last slide of introduction before I jump to the introductory part. What this table here is supposed to show you is kind of all the necessary steps in columns regarding a clinical performance study. So in short, this is going from just study design and planning the study, writing that down in a clinical performance study report, then before starting, actually getting ethical and legal clearance for the study, procuring the samples, including gaining partners and recruiting donors, managing the data, evaluating it, and then writing everything together in a clinical performance study report. So this is just one singular process, and all of that also is covered by our services, by the way. But what I would like to stress here, rather, is not that, but the fact, which you can see in the percentages at the very bottom of the table, is that the workload that is required during the process of this study is not evenly distributed. On the contrary, there is a strong concentration of the necessary workload, which means both time and costs and manpower, in the middle of the study. And those are all the steps that have to do with sample procurement, gaining legal clearance, finding partners and laboratories, donation centers that could work together with you or that fit for the study purpose, finding the necessary donors, monitoring everything, and analyzing these samples in the correct fashion. That is what is where a lot of money and a lot of time is involved. So at the above this diagram, where you see the average time and lead costs of approximately half a year, approximately 150K, this is very much dependent, these numbers. So these numbers are very rough. Please don't take them as absolute estimations, but rather very high level estimations. These numbers are mostly dependent on those 80% at the bottom of this diagram, which means how many samples do you need? What kind of samples do you need? What kind of analysis do you need of these samples? These are the questions that are decisive for the size of the project you're talking about. Now, with this in mind, I would like to start with the actual informatory part of my talk, and to introduce this. The major part of the technical documentation that an IVD manufacturer would need to submit in the EU at the notified body in order to gain a CE mark are the performance evaluation plan and the performance evaluation report, which are basically including what do you have planned on generally doing to evaluate the performance of your device and what was the outcome. And in between this, there are the well-known three aspects of scientific validity, analytical performance, and clinical performance. So if you would like to see it that way, like three branches or three columns of performance evaluation that are made up of these three factors. And now for the first two, the scientific validity and the analytical performance, there's a lot of information and help out there on how to do that. For the scientific validity, you would just follow through with the official guidelines of a systematic literature review. That is an official defined way of doing literature search in a way that you have reproducible outcomes. And for analytical performance studies, there is a plethora of CLSI guidelines and other guidelines out there that tell you exactly what you need to do in order to show limit of quantification, linearity, high dose hook, and all those other things for analytical performance. For clinical performance, however, there is no such thing, or if yes, in very rare cases only. So the question that I, from the point of a CRO, mostly get is, how do I do the clinical performance studies? How much does it cost? What do I need to look after? And first thing, don't look in the IVDR or the ISO 29116, which is the major ISO for anything regarding clinical performance studies, because none of these documents will give you like any details on what to do. They tell you do it right, but they don't tell you what exactly to do. However, at least something, the IVDR is already like foreshadowing that there will be common specifications that will help with the matter a lot. And indeed, under the IVDD era, we had like the common technical specifications for list A tests. So that was HIV, hepatitis, and everything. And exactly these products have been 2 years ago already implemented or included into a new IVDR-compliant common specification document, which is out there. However, a lot of you also know these common specifications because anybody who does or who manufactures tests around HIV and hepatitis, of course, need to follow these. But apart from the 12 or 13, I believe it's 12 IVD products or kinds of IVD products included in the common specifications, there is basically no standard out there for anything. So common specifications are the absolute exception. Only for 12 products so far, there are distinct common specifications. So what do you do if you don't have common specifications? Which means for basically any other IVD product, how would you approach the problem of study design and doing a clinical performance study? Now we'll drop a term here for which you can basically fill books with the definition of this, which is the term state of the art. Just quite generally, or very, very shortly, because again, this is a very large matter, but state of the art from the point of view of the IVDR means anything that currently, at the time of where you want to introduce your product to the market, is like general practice in the field you're working in. How is are diagnostics happening? What is the clinical practice right now? What is your target marker? How is it behaving currently? All those things are, of course, are dynamic, that can change over the time of the course, but it's always is considering the exact time of where you place your application. And let's say a decisive parameter that defines on what kind of state of the art you need to fulfill is the intended purpose of your product. The intended purpose is basically one of the most important parts of how you define your product. The intended purpose is supposed to be, according to IVDR, pretty much the first sentence of the instructions for use of your product. And it will define the assay type, it will define the technology that you use, if it's automated or not, what is the intended user, in what medical field you're working in, is it quantitative or qualitative? All this information is supposed to be in the very first phrase of the instructions for use, and that is put together or summarized as intended purpose. And I can't stress the meaning of this word enough. This really defines everything after. So if you think about designing a study to validate your specific product, looking at the intended purpose already helps you like envisioning how the study must look like. If you say you have a product that is a point of care product and is supposed to be used by the patients themselves, maybe so a layperson use product, it would be completely wrong to just say you go to a clinical laboratory and validate the test there just because they have like leftover samples. That would not be the correct approach. You need to look at what kind of study site do you need, what kind of comparator do you need according to your intended purpose. If it's a point of care device, design the study that you envision the study. Okay, the study needs to take place at the point of care, at the bedside test. That's how I need to organize it. Apart from just, and also that was just a glimpse, of course. I lack the time, unfortunately, to teach study design. That's also something that can fill books. But with that in mind, a main concern still remains always, even if you know how to design your study and like where it needs to be, who needs to be the users, etc. The main concern will stay the samples, which comprise the 80% of time and costs that I showed you in the table just a minute ago. And question number one, when thinking about samples for your study, would of course be, how many do I need? An easy answer to this is, of course, check for common specifications. Right now, again, we only have one common specification document. In the future, there will be more, hopefully. But if there are common specifications, just use them. It's simple. There's no questions asked. If you say to the notified body, you followed common specifications, that you can show it, you are compliant with IVDR. It's very, very simple. Usually, though, that is unavailable, and you have no other way out in that case than delving a little bit into the field of statistics. However, also here you have kind of shortcuts, so to say, that you can take, which are also viable. First of all, of course, statistics is complicated, and nobody really wants to deal with that except for statisticians. Very simplified. However, you, like, from a manufacturer's point of view, you need to only know 4 parameters in order to do a first level of sample size calculation. So calculating how many samples you need. And that would be, first of all, your desired sensitivity and specificity. So those 2 parameters, like what do you intend, or how sensitive you intend your product to be. You need to know that even before the study. Yes, it's a little bit of a paradoxon. You need to know how secure you want to be with that estimate. So either how wide would the confidence interval be in this case, or alternatively, what do you want to set as the lowest border of acceptable sensitivity and specificity? And lastly, but also not even in all cases, you need to at least have an idea about the prevalence of the disease in your target population. So nothing that you need to like completely understand currently. It's just that with these informations, you can do something very, very convenient, which is checking just official scientific literature, 2 of which I've quoted at the bottom right of the slide, which include tables for certain combinations of these parameters, which give you pretty exact numbers of sample sizes of what you would need to analyze in your study to be IVDR compliant. After all, for instance, just an example, you have, say, and introducing also the term state of the art once again. Say you have an ELISA which you need to be at least as good as your competitor's product, also an ELISA that has a sensitivity of, let's say, 85%. And you now need to know, or would like to know, how many samples do you need to prove, so to say, that your test has at least the same sensitivity. And you say you are probably around 90% sensitivity. Then you would look at this table and at the y-axis go down to 0. I don't know if my cursor is visible, but would go down to 0.9 of expected sensitivity, and then follow the table to the right on the x-axis to, at the top here, the minimal acceptable lower confidence limit, which would be, in your case, the sensitivity of your competitor's device, so 85%. And then you could just read here, all right, I need 474 samples for, and that is positive samples then, for the proof of non-inferiority of my sensitivity compared to my competitor. So you don't need to reinvent the wheel, especially not when it comes to statistics for your clinical performance study. There are publications with tables that can really simplify your life with this. Yeah, this state of the art is exactly giving you the example that I just quoted. Think of what you need to achieve from the view of the state of the art, then take these parameters to check on these tables what the sample size for your study must be. So if you want to do it like 100% perfect, sample size calculation is a science in itself, but that is not necessary. It is absolutely sufficient to just use these tables if you know how to have a rough, let's not even rough, it's pretty precise. It's a pretty precise estimate of the sample size that you need, and you can just use that. It's enough of a rationale. Question number 2 regarding samples would, of course, or question number 1 was, how many do we need? We have that answered. Question number 2 is now, what kind? And now we are again pretty straightforward back to, first of all, if there are common specifications, just use them. And if not, it's pretty much following the same principle as with the study design in general. What is the state of the art in medicine, and what regarding this is the intended purpose of your product? So If you claim your product is usable with blood, serum, and plasma, obviously you need to include blood, serum, and plasma in your study. If you leave one out, you cannot make that claim in your IFUs or in your marketing material. If you have an influenza test and you know that pretty much annually you have a new mutant, a new variant out there, you cannot just use 5-year-old samples. That will not suffice. You need to include the actual recent variants. If your product is used for instance, pathogen distinction, is it influenza or is it COVID? Then you need to include samples from both of these diseases or also other similar diseases to say or to prove the claim that your product can actually make this distinction. From an epidemiological point of view, if you do an allergy test for pollen that is abundant in between March and July, do not design the study to start in October. Very simple thought, obviously, but I see this done wrong or forgotten, omitted in the planning a lot of times. That's why I would like to stress this. Okay, before we go there. Now, these are all very simple thoughts, but all of that in combination require a lot of concentration that you need focus on including all of this in your thoughts on study design. And with all these little building blocks, so to say, on your sheet, what you finally need to decide on is what kind of general design you want to go with your study. And these are the last two slides of this presentation from the informatory view. Because you can do the study retrospectively and prospectively. And I know these are very, very often used terms, but I am under the impression that there's not complete understanding on what this actually means. So I would like to go through this really quickly for both of these designs. First of all, retrospective design means like simplified, which means that the inclusion criterion for the samples is a diagnosis that is given for the samples. Since I get the question a lot whether this is even still allowed under the IVDR, so is it okay to use retrospective samples, leftover samples? Just a quote from the ISO 29106, which claims that most studies for IVD medical devices are performed using samples resulting from the remnants of specimens taken for purposes of standard of care. This is a long phrase, which just means usually studies are done using leftover samples, which basically means for you, leftover samples is completely fine. You can use leftover samples, however, not in all cases, at least not without taking risks. More on that in a minute. So, an important factor for sample size for you regarding the sample size is that if you do a retrospective design, you completely ignore the disease prevalence. It doesn't matter if the disease is rare in your target population. If you know you need 100 positives, you just take the 100 positives from people you know that are positive. So it doesn't matter that it is rare because you targetedly collect the samples from positive individuals anyway. So yes, you targetedly look for patients with a certain diagnosis. He or she is positive for a very specific type of influenza, for instance, and you take the samples from them, ethically, obviously. So that means you take a certain amount of positive samples for estimating sensitivity and a certain amount of negative samples for estimated specificity. And you're done, basically. So for a very high level perspective, that would be enough to calculate sensitivity and specificity of your device. However, and this is of course known to the notified bodies, going this way, you have a very high risk of overestimating, the performance of your test. Which is the reason for that is simple. You usually in nature, like in the real setting of using your test, you don't have this nice distinction of these two groups, positive and negative. There is a lot of in between. And if you ignore that in your study, you get a much higher sensitivity and specificity than your product actually has. So what you need to do in a retrospective setting is always including carefully designed distinction panels. Like I just said regarding state of the art, if it's an influenza test, include para-influenza, include COVID, include all kinds of bacteria and viruses that would cause similar symptoms, because distinction here is the absolute key for the intended purpose of your product. Keeping this in mind, you need to design in a retrospective design at least some sort of panel that alleviates the problem with overestimating the performance. So you need another cohort, so to say, of samples from patients with similar diseases. On the opposite side of the possibilities of how to design your study is the prospective design, which is frowned upon by manufacturers, I feel, a lot. I usually get asked for specifically for a retrospective design, but the prospective design has a lot of benefits, which I want to show here, or at least especially one major benefit. First of all, just quite generally, prospective design means that you don't take the diagnosis of a patient for including them in the study, but rather the medical indication, that of course is the medical indication of your product, to include that person in the study. That means that when you include somebody in the study, you do not know if the person is negative or positive. You just know the person has a certain set of symptoms, for instance, coughing and fever, for example, and you don't know if it's COVID or if it's influenza. And this is in line with the IVDR, actually, which very distinctly says that where appropriate, the performance study that we talk about here shall be performed in circumstances similar to the normal conditions of use of the device. And staying with the example, if you have an influenza test, usually the conditions of the device would be in a setting that a patient comes into a doctor's office with certain symptoms, and the doctor wants to know what kind of virus is causing the symptoms. So, and that is one of the really cool things of this design is you use the actual target population of your device as the study population. This also means that you need to include the disease prevalence, which means that you usually have much higher sample sizes, which also increase the time and cost of a prospective study. Not always, but usually. But it has its advantages. It also means, for instance, that you only have one cohort. You only have one amount of samples, one number of samples in total, which you calculate by including the disease prevalence in your sample size calculations, which includes both positives and negatives. So all patients you need, or all samples you need for sensitivity and specificity estimation. And now the major advantage of this design is that any borderline cases, any fringe cases, anything that could decrease, so to say, the performance of your test, which is not a bad thing. It's just it's a real setting, so to say, is automatically included in the study and in your study population. You don't need to include any additional cohort which you artificially put together with similar diseases, or say, if you go by the medical indication as inclusion criterion, so in a prospective design, you have these people automatically included. This also means, however, that usually archived samples are not viable, because for this kind of design, you need information regarding the medical indication of collecting the sample in the first place. And for archived samples, this information is usually simply not there. It's not collected, if not specifically for a study, and therefore, even we, with our dense network of partners, usually don't have access to archived samples with this depth of information behind it. So you need direct cooperation with medical institutions and CROs that know how to do that here. And again, the lead time and the costs are a little bit higher than with in a retrospective design. Usually you could estimate, let's say a retrospective design would be 140K or 140,000 euros. Then a prospective design could be between 170,000 and 200,000 compared to the retrospective design, just as a rule of thumb, very rough estimate. Also a very important prospect is that we expect this design, the maybe more complicated, actually not, but it seems more complicated prospective design, to become the preferred model by secondary legal acts in the future. And we'll have to see how the notified bodies, of course, approach this problem. We have seen this already during COVID pandemic, during COVID in the EU, COVID antigen rapid tests that had prospective performance studies behind them, were preferred for reimbursement by the authorities, which pretty much meant that COVID tests with retrospective studies behind them were out of the picture. So very important to know, of course, for manufacturers. And we expect similar things to happen in the future. And now, very, very simply, like as a take-home message, there was a lot of grayscale in what I just described for you. How many samples to take? You have to think of the state of the art. You like have a lot of leeway on where to go exactly with your study design, with how to define your sample size and how to define your sample types. But whatever you do, have a rationale. And also, if you have a rationale, a good one, you're basically safe. If you say, we designed the study on this basis, which for us made the most sense to reach a condition similar to the normal conditions of the device. And yeah, usually the notified body, of course, they can say, I don't believe you, or I have doubts about this. But if you have a rationale, you always have a foundation to which to fall back to, and you have reasoning for what you did. And if you don't, the notified body will ask. So have a rationale, whatever you do. And very lastly, because also this like especially for our Chinese friends, understand that this time right now with the IVDR being new is of course complicated, but it's complicated for everybody. Right now, there is barely any products on the that are IVDR compliant CE certificated, because European manufacturers are also not moving. European manufacturers have the same problems as anybody else. Everybody is in the same position, and it wasn't like that before, which means there is a gap, there is a vacuum in the market right now. See this. Even the big players, like all the big shots of the industry, they're not doing as much as you would think. There's an immense demand for certifications, for applications right now. And notified bodies have also free capacities right now. It's not as bad as a year ago. It's actually quite fine at this moment. Take this opportunity to gain market share. The European patients will thank it to you a lot, which is, we forget sometimes the reason all of this exists and our whole field of industry exists. And with this, I would like to thank you very much for your attention. I hope everything worked out from a technical point of view. These are the faces that are doing anything for you. The gentleman in the middle in the blue suit and the brown shoes is like the head behind everything that in.vent does, Professor Hollett, a genius of the industry. So don't hesitate to place your trust. And thanks a lot for your attention again.